Okay, good evening, everybody. Let's start our today's session, Medicinal Chemistry, Week 7, problem solving session. Okay, is my screen visible?
Yes, it is.
Okay. Okay, so today our first question is about a true or false question. So it is saying that the term drug tolerance is used to describe the effect of a drug that diminished after repeated dose of administration. So is this true or false? Anybody? Am I audible? Am I audible? Somebody please respond, otherwise I cannot understand whether am I audible to or not.
Yes, sir.
Okay. So, is this true or false? The drug tolerance, what is described in our question. Okay, so let's first discuss what is tolerance. Can anybody tell me what is the drug tolerance mean? In simple language,
Sir,
what do you,
sir, drug tolerance when increase the dose, but it's not give the biological effect.
okay, and what is the
This word
cause?
is
This is the effect, and what is the cause of drug tolerance? When we
drug
use a
tolerance is
drug very much repeatedly.
bioavailability. Bioavailability is completed. Is taken over all the receptors as complete dose. Receptors as
Yes,
saturated.
yes, yes. When we take a drug very much repeatedly or in multiple times, then our body gets habituated or saturated with the drug. Then we have to increase the dose for our effective therapy, right? So tolerance is body's diminished response to a drug, which occurs when a drug is used repeatedly and body adapts to the continued presence of a drug. What I have said that when we use a drug very repeatedly, then body get used to or adapts to the drug, continued presence of the drug. So this is where we can explain the drug tolerance. So our statement is true or false. It is true statement. So, a person may develop tolerance to a drug when a drug is used repeatedly. For instance, when morphine or alcohol is used for a long time, larger and larger dose must be taken to produce the same effect. Usually, tolerance develops because of the metabolism of the drug speeds up, often because of the liver enzyme involved in metabolizing the drug becomes more active, and because the number of the site cell receptors that the drug attaches to the strength of the bond between the receptor and drug decreases. Okay, so now along with tolerance, another term comes with the tolerance, that is resistance. Can you tell me what is the definition of resistance?
The drug doesn't produce any response.
Yes. So, what is resistance? Resistance refers to the ability of the microorganism or the cancer cell to withstand the effect of the drug usually effective against them. So this is called the resistance. So strains of microorganisms, generally bacteria or viruses, are said to develop resistance when they are no longer killed or inhibited by the antibiotics or antiviral drugs that are usually effective against them. So, so, or in the practice when significantly higher than the normal dose are required to have the effect. Similarly, cancer cells may develop resistance to the chemotherapy drug. Resistance appears because of the mutations that take place spontaneously in any of the growing microorganism or the cells, whether exposed to drugs or not. Most such mutations change the microorganisms or cell structure or biochemical pathway in way that is harmful to the microorganism or cell. But some mutations change the parts of the microorganism or the cell that are affected by the drugs, decreasing the drug's ability to work. So, what is resistance? When we are using an antibacterial or anti-cancer drug against some microorganism or against some cancer cell, so it is the ability of that microorganism or that cell to withstand against that drug. So more and more dosage is required to produce the same effect. So this phenomena we call it as the resistance. Okay, so this is our tolerance.
Sir, I have a question. Like,
And
if only microorganism has the resistance, or receptor may also have the resistance regarding the drug?
no, microorganism is not getting affected during the drug. They are mutated in such a way, or they are continuously mutation is going on in case of bacteria.
But
So
but
they mutate their
in
cells.
in case of diabetes, when taking the continuous dose, then drug is also resistant. So there is no microorganism. I think then is the receptor is
Receptor is also involved. Yes.
yeah, then receptor is involved for the resistance.
No, that is also a case of the tolerance. Whatever we discussed earlier, that in case of the in kind of disease, when we are taking a dose for the longer time for any disease, so the receptors get habituated to that particular drug or that chemical. So that is the case of the tolerance. And resistance in the we differ that when some microorganism or some cancer cell uh, are involved and they can be mutated against that drug, we are referring that as the resistance one.
But in resistance, if we increase the dose, then effect will come.
Yes, it may come. It
And
is also,
in tolerance, if we increase the dose,
yes, that is also the case.
then what is the difference between tolerance
It
and
is the
resistance?
difference between some microorganisms are involved in case of the resistance. They are become resistant. Okay,
Okay,
and this is the effect of the cell receptors.
okay.
Okay.
Okay, but I have confusion that so many doctors says that in case of diabetic the
Hmm.
drug is resistance now, so we have to increase the dose.
Yes, those terms are related one, very much related, but there is a difference between these two. That is why I have mentioned the resistance one. Here, resistance is not at all mentioned in the question, but that is
Okay.
why I have discussed about the term resistance.
Okay, so resistance term related to the microorganism.
Microorganisms or cancer cells, like they
Okay,
are getting resistant against some common drug or common therapeutics.
okay, okay. Thank you, thank you.
Okay, so can we move to the next question?
Yes, sir.
Okay. Okay, so it is saying another true or false question that drugs are taken orally, pass directly to the liver once they enter to the blood supply. This is known as the fast pass effect. It is true or false?
Yeah, true, sir.
Okay, true. So, can anybody tell me about the fast pass effect? What does it mean in short?
Sir, when the drug goes directly to the liver, not is to the giving this in through gut or systemic circulation, it directly passed to the liver. Then it
Hmm.
caused the first pass.
Hmm. So, whenever we are taking a drug orally, then it has to go through the pass through the liver. So, what it happens, it is a phenomena in which drug get metabolized at a specific location in the body that results in a reduced concentration of the active drug upon reaching its site of action or systemic circulation. So, not only liver, the most of the major place is the liver, but it may also be passed from the GI tract or the gut wall. So majority of the drug fast pass effect is related to the liver. So that is why that liver is very important in case of the fast pass effect. So what it is saying, that the fast pass effect is a phenomena in which a drug get metabolized at specific location in the body that results in a reduced concentration of the active drug upon reaching its site or systemic circulation. The fast pass effect is often associated with the liver as this is the major site of the drug metabolism. However, the fast pass effect can also occur in the lungs, vasculature, GI tract, and the other metabolically active tissues in the body. This effect can become augmented by the various factors such as the plasma protein concentration, enzymatic activity, and gastrointestinal moiety. The extent to which a patient may experience the fast part effect varies from patient to patient. So the amount of the drug is metabolized at the liver is depending upon the particular patient to another patient, and this must also be taken into consideration when determining the appropriate dosing. This is very obvious that for a particular patient, if suppose 10% is metabolized, and for the another patient some 20% or 30% of the drug getting metabolized, then definitely the dose of that two patients will be different. Okay, if the fast pass effect is exceptionally prominent in a patient, the drug may require administered via a different route to bypass the fast pass effect. So is this clear what is written here? The last line is very important. So whenever we are taking orally drug, so it is uh, passing through the liver, right?
Sir,
If
I
the,
have doubt.
yes.
If it metabolizes in liver, then we can say it is a first pass effect. But here not mentioning the drug it metabolize, it just saying that it goes to the liver.
Uh, Once again, drugs that are taken orally pass directly to the liver once and they enter blood supply. So, what is your doubt?
They are not mentioning the drug metabolized in liver.
No, it is not mentioned metabolized in liver. So, it is telling na, the drugs that are taken orally pass directly to the liver once they enter to the blood supply. So, after liver, they are entering in the blood supply. So, definitely, it is the fast pass effect. Na? So there are notable drugs that get significant fast part effect are insulin, morphine. Okay, so liver metabolizes many drugs and sometimes only a small part of the active drugs can go to the active site. Fast pass effect of the liver may lead to the low bioavailability of the drugs. Okay, so our this statement is also true. Okay, so another question is about what is meant by a lead compound in medicinal chemistry. So what is a lead compound? A drug containing the element lead or a leading drug in a particular area of medicine, a compound that acts as the starting point for a drug design and development, a drug which is normally the first to be prescribed for a particular.
Sir, C. A compound acts is the starting point for the drug design and development.
Okay, so what does it mean, the lead compound?
So lead compound is the substance by which we can start our research, or we can think about that we can produce a real drug.
Okay, yes, this is absolutely true. A lead compound is a compound that acts as the starting point for drug design and development. It may also have some disadvantage like some low activity or the presence of the side effect, but it acts as a starting point for development of a particular agent. Okay, so what it is saying that in medicine, a chemical compound that shows a promise as a treatment for the disease and may lead to the development of the new drug. Thousands of the compounds are tested in the laboratory for to find a lead compound. Lead means leading. The compounds which can leading to the new therapeutics. Compound that may act on the specific genes or the proteins involved in a disease. Once a lead compound has been found, the chemical structure is used as a starting point to make the drug that has the most benefits and least harms. Okay, so it is the basis of our development of a new drug, that is called the lead compound. So finding a lead compound is the first step in making a new drug to treat a disease. So with that lead compound, another term comes with this term, that is lead optimization. So you have any idea regarding the term lead optimization? So from the name itself, what we can understand that, as we have seen or discussed, that the lead compound has some disadvantage, may also have some toxicity or low bioavailability. So lead optimization means the optimization of that particular structure. Okay, a lead compound has been identified and now must be optimized to become a candidate drug. Lead optimization leads to designing and improving a pre-identified lead compound and involves manipulation of multiple parameters of the compounds. In addition, the compounds need to follow the ADMET property. So a lead is the starting point, as you have said, that from that we are starting our development of the drug. So suppose it has some disadvantage, suppose it has the low solubility or the toxicities, have some kind of toxicity or bioavailability issue. Bioavailability issue is it may have some lead compound. So in lead optimization, what we supposed to get, we have to optimize those points here, that we have to develop or introduce, suppose, a group which can increase the solubility of our compound, or if it is showing some sort of toxicity, then we can optimize or modify our compound in such a way that our toxicity is less in case of the in vivo and in vitro study. And if bioavailability is low in our lead compound, then we can such a way modify our structure, it can increase the bioavailability. So these terms comes under the lead optimization. Okay, so from lead compound, it is our answer that a compound that act as a starting point for drug design and development, and lead optimization refers to that modification which can be optimized to make a perfect candidature for a drug molecule, a marketed drug molecule. Okay, is this clear?
Yes.
Okay. Okay, then let's move to our next question. Okay, so which of the following statement is false with respect to the NMR screening to detect the drug target interaction? So, which is not false. So, there are some statements given. The process relies on the small molecule having shorter relaxation time than the large molecule target. Then it is telling that the process can be used on mixture of the compounds. The method can detect weak binding. The method can identify small molecules binding to different regions of the same binding site. So, so what it is saying that the process relies on the small molecule. The we have already discussed that small drug binds to our receptor, right? So, which one is smaller, drug or receptor? When a drug, it is maybe a small molecule or any kind of natural product, when it is bind to a receptor one, so which one is the smaller with respect to the molecular weight, this drug one or the receptor?
Drug.
Drug.
Definitely the drug. This is have the very low molecular weight, and the receptor, it is a protein somewhere very macromolecules. So this is definitely having a larger molecular weight. Okay, so it what it is saying, the process relies when we are detecting this binding kind of things through NMR, then small molecule, that is the drug one, this drug one having longer relaxation time than the large molecule which is target or receptor, this one. Okay, when a small molecule drug binds to a macromolecular target, it essentially becomes part of that target, and the signals for that compound will have the shorter relaxation time. So, if there is a time delay in measuring the NMR spectrum, the signals for the molecules that bind to the protein will disappear from the spectrum. So, whenever a drug is completely bind with the receptor, then it is definitely a part of that receptor, then it must show some shorter relaxation time, and if there is some delay in measuring the NMR spectrum, the signals of the molecules that bind to the protein will disappear from the spectrum. So our this one is the false one, the another three are the right one. So this is our desired false statement. So now in this next question, what we are getting that there are several sources and methods of discovering new compounds. Okay, so it is asking that which of the following is most likely to lead to the discovery of a complex structure quite unlike any other previously discovered. So can you tell me what is combinatorial theory? sorry, combinatorial chemistry. Any idea, anybody?
More drugs can be synthesized. More similar drugs can be synthesized.
Yes, it is involving the wet lab synthesis of a large number of the compounds, right? Okay, so combinatorial chemistry involves the synthesis of a large number of the compounds using a standard synthetic route, and so the types of types of structure that will be synthesized are predictable and belong to the similar family of the compound. So a similar kind of synthesis we are getting in the combinatorial chemistry. So, okay, so similar kind of other some molecular library we are synthesizing in our lab. So this is saying a combinatorial chemistry, right? So what is database mining? Anybody? From the name itself, we can guess some idea regarding the database mining. What does database means? Anybody?
Large number of data.
Yes. So what does database mining mean? So what is there in the database in our with respect to our question? Okay, so database is basically a storage of the data. It may be a different kind of data. So in respect to our medicinal chemistry, we are as we are dealing with the drug or some kind of medicine. So definitely we will deal with some this database of the medicines. Okay, okay, guys, can you get me a 5 minutes? I have to pick up a call. It's urgent. So can I take one just for 5 minutes? I will come back.
Yes.
Okay, thank you.
Yes, sir.
Okay, sorry guys. Am I audible?
Yes, sir.
Okay, so we are talking about the database mining. So in case of the database mining, various kind of databases are there, like PDB, protein data bank. Okay, so like PDB, so there are various kind of protein structure which are stored in that data bank. Like this way, some data bank mining is also consisting some kind of drugs part related to the particular disease. So basically, this is a computerized screening of various large number of the compounds. So in that way, we can also search for the new type of the compounds. And the fourth one is the Me Too drug. So can you tell me what is Me Too drug mean? What does it mean?
The structure is the same drug which
Yes,
we know
yes,
for the same function.
yes, exactly. The Me Too drugs, the originally there was a pre-existing drug, and the Me Too drug, new one, is a slightly different or very much similar to the older drug. So, and the screening of the plant extract, this is the one. So, how can we extract the plants? Okay, so as we know that natural product which are basically from the plant source is a very good or very much rich in source of the medicinal drugs or bioactive materials. So these natural product which are isolated from a tree extract is very much important in case of the medicinal chemistry. Okay, so this is a very good technique for developing a new kind of drug. What we do usually, that if we get a mixture of the natural products from a plant or some any kind of origin, so our next target is to separate that drug or some bioactive compound what we get through the column chromatography. Okay, so after the separation of this column chromatography, we are getting a individual compounds. Then we do the structure elucidation via mass or NMR or HPLC or elemental analysis. Just a second, guys. Just a minute, 5 minutes. Just give me. Sorry for the today's interruption. Okay, so sorry for the interruption. So from these three or four techniques, what we discussed here, that is combinatorial chemistry or the database mining or Me Too drugs or screening of the plant extract. So what do you think that whichever is the most likely way to optimize the lead discovery of a complex structure, quite unlike any of the previously discovered?
Screen of the plant extract.
Yes, exactly. Screening of the plant extract, because there are no preconceived ideas when screening plant extract for the active lead compounds, because these are the completely new kind of things we are getting from a plant extract. So we these are completely new compounds. We have to discover or elucidate the structure and the activity of these compounds after the actual separation. Okay, so the other kind of drugs like me too drugs, it is as we have discussed that it is somehow similar to the pre-existing drugs. And database mining, we are dealing with the drug which are already present in that data bank, right? So this is the not a very new idea to discover the new drug. And combinatorial chemistry, as we have also already discussed, that we are trying to develop a new similar kind of chemical library which is already existing as the accepted drugs. So the screening of the plant extract is the our answer which can be used for the discovering new compounds. Okay, so which of the following drug was not isolated from a natural source? Okay, let's come one by one. Quinine, where from we get the quinine?
Cinchona,
Cinchona. And what it is used for?
antimalarial.
Antimalarial drug. Okay, very good. So quinine is isolated from the bark of the cinchona tree. So it is anti-malarial drug. And then morphine, where from we get
Opium
it?
poppy, obtained from opium poppy.
Yes, seed pods of the poppies. Okay, so morphine is also getting from the plant. And then this one, artemisinin,
Yes, it is also antimalarial drug obtained from the plant.
yes, obtained from the plant. So whichever is left,
Isoniazid is a totally synthetic one.
totally synthetic one. We synthesis in the lab one, right? Synthesized in the laboratory. So this is our desired answer. So it is clear to everybody. Okay, so we are moving to our next question. Okay, so here it is saying that we have already discussed earlier, the term me too drugs is used for the drugs that are similar in structure to a known drug and which are used for the same purpose. So this is true or false?
True, true.
True. Yes. So let's see some kind of, if we can discuss the example of the me too drug. So let's consider two structure. So can you spot the difference between these two structures? They are very much similar, but there is a certain difference in two structures. Can you spot that?
In one drug, there is a pyridine ring.
And another, you are telling this one, this one.
Yes.
No, this is in both the cases. This is also pyridine ring, substituted pyridine. Okay, so what we define as the Me Too drug? The Me Too drug as a pharmacologically active compound that is structurally related to a first-in-class compound that is already established, regarded as belonging to the same therapeutic class as the original compound and used for the same therapeutic purpose, but which may differ in some respect, such as a specificity of the pharmacological factor, adverse reaction profile, or drug-drug interaction. Means they are totally different. The active moiety is completely same with the already established drug, but there is little bit difference within the structural activity or structural of that previously established drug. So that drug we consider as the Me Too drug. Okay, so this statement is definitely true. So you could not identify the difference between these two structures. There is a fluorine is there, fluorine atom is there, but here it is not present. Okay, so as you can see that large structure, this long chain structure is completely same like the previous one, but only in very specific position some difference, a little difference of the chemical structure is there. So this is why it is calling as the Me Too drug. Okay, so we are moving to the next question. Okay, so which of the following is the correct definition of the bioavailability? So what is bioavailability?
Amount of drug present in systemic circulation.
Yes, it is saying the availability of the drug or the other substances to be absorbed and used by the body, right? So orally bioavailable means that a drug or the other substances that is taken by the mouth can be absorbed and used by the body. So whatever the amount whatever we are taking orally, or we are putting in injection in the IV or IP injection, so whatever the amount it is getting absorbed for its actual action in the body, we are terming as it as the bioavailability. So whatever how the amount is available for its function, right? So let's see what it is saying, that bioavailability describes the portion of the drug administered that is metabolized very quickly and thus it is not available to induce a physiological effect. Is this true? The first option?
No.
No, definitely it is not. If it is very quickly metabolized very quickly and that it is not available, it is already mentioned So how can it be regarded as the bioavailability, right? Okay, so the next one is saying that the bioavailability describes the ability of the administered drug metabolized to cause a undesirable physiological effects. Is this true?
Yes, it is not true.
It is not true. So the amount bioavailability describes the ability of the administered drug metabolites to cause the undesirable physiological change. So definitely this is not bioavailability when it is dealing with the undesirable physiological effects, right? We are dealing with the its positive effect, how much it's getting absorbed to show its desired effect, right? Is this clear?
Yes,
So
sir.
it is
Yes.
saying that the bioavailability is the length of the time an administered drug is present in the body and thus is available to cause a physiological effect. So is this true? Is this
No,
dependent on the
it is
time
called duration of action.
duration of action? Yes, very good. So it is also false with respect to bioavailability. So the left option is bioavailability is used to describe the fraction of the dose of the drug administered that is present within the body and facilitates the desired physiological effects. So this is the our right option. Okay. So, bioavailability of a drug is linked to the route of administration as is dependent on which epithelial tissue are involved in the absorption process. Without appropriate administration, the subsequent absorption of a drug may lead to the suboptimal concentration and distribution throughout the body, resulting in no or inappropriate physical response. So by which route we are applying it, bioavailability is also dependent on that. As we have also mentioned the fast pass effect, we have also discussed that when a drug if it's getting metabolized within a liver, then its bioavailability will be low or high. If it is getting metabolized in liver,
Low.
low. So if it is getting at the metabolized, then a very lower amount will be reached to the circulation or to its active site. So then we have to choose a different kind of administration process if we cannot put it orally, if bioavailability is very much less in case of oral administration. So definitely we have to the route of administration is very much important in case of the bioavailability. Okay, so that is why it is mentioned that without appropriate administration, the subsequent absorption of a drug may lead to the suboptimal concentration and distribution throughout the body. So the route of administration is very important in case of the bioavailability in determining the bioavailability of a active drug. Okay, is this clear? Can we move to the next question?
Yes.
Okay. Okay, so which of the following integral membrane intrinsic protein facilitate the transport of the water between cells? So what does it call? It is rhodopsin or glycoporin or cadherin or aquaporin.
Aquaporin.
Aquaporin. And what is aquaporin?
which are having a six member, and there are small pores and which are helpful for the drug transportation.
Drug transportation, and the major what it is, that it is water channels.
Yes, water channel.
Water channels. Aquaporins are also called the water channels, are the channel protein which across the cell membrane, right? It is the transport of the water across the cell membrane in response to the osmotic gradients created by the active solute transport. Okay, so it's maintain the osmotic gradient within the cell. Okay, so is this clear? So aquaporins, also called water channels, are the integral membrane protein from a large family of the major intrinsic protein that facilitate transport of the water between cells. Okay, okay. So our right answer here is aqua. So let's move to our next question. So now tell me, what is the term used for an animal that has been genetically modified for in vivo test? First, you tell me what does in vivo test means.
In vivo test means inside the body.
Inside the body or inside the living system, right?
Yes, yes.
And in vitro means,
Outside the body, maintaining the all temperature and all that which are present
yes,
inside the body.
means cell culture, right? We are mimicking the type of the cell in the laboratory, external to the body. Outside the body, we are maintaining the exact condition of our body. That is in vitro study, and the in vivo is within our living system. Okay, is this clear to everybody? In vivo and in vitro. Everybody, is this clear, this term in vivo and in vitro, to everybody?
Yes, yes.
Okay. Okay. So, what are the term used for the animal that has been genetically modified for an in vivo test? So, what is the answer?
Transgenic animals.
Transgenic. And what does transgenic means here?
In that we made some changes as per our need.
Yes. In case of for our experiments, we do the genetic modification, right? Knock out the gene. Like suppose we are testing on some neurological behavior test, so we need a Alzheimer model mice. You know about what is Alzheimer's? Okay, this is a very common form of dementia, and this is very common
Loss of memory.
yes, neurodegenerative disease, loss of memory, right? So suppose we are testing for in case of the Alzheimer disease, in case of the rodent model, in case of the rat. So what we do to check the activity of our compound, we can use the transgenic mice. That is the particular gene which is responsible for this disease. We are modulating genetic modification of that disease so that it can induce that particular disease or that particular model. Then for the our study we are using this kind of animal. So this is called the transgenic animal. So this is very common in case of the our in vivo studies. We it is variously used, transgenic animal. So moving to our next question, which of the following is not classed as the in vivo test? So first one, it is showing that the testing anti-cancer drug on a tumor grown on the back of a rat. So is this in vivo test? Yes or no?
Yes, it is in vivo.
Why?
Performing inside rat.
Performing inside the living rat. Okay. So then second word is testing analgesics by determining guinea pig sensitivity to pain. Is this in vivo or in vitro?
In vivo.
In vivo. Because it is also using on the guinea pig, right? And testing antibacterial agents in infected rats.
Yes, in vivo.
In vivo, as it is used in the infected rats antibacterial agents. And so this one, testing antibacterials by measuring the growth of the microorganisms on a petri dish in their presence, this is
In vitro.
in vitro test. This is in vitro because here no living animal is getting used. Here we are getting on the culture plate, right? In the petri dish, we are giving growth to the microorganisms and then we are testing our antibacterials on that petri plate, right? Is this clear?
Yes.
Okay, so in vivo test is carried out on the living systems such as the animals or the human volunteers. So first it is getting tested on the animals, as you know, that after various trials it is coming to the human. And in vitro test is carried out on the target molecule, whole cells, isolated tissues or isolated organs. Microbiological cultures are considered to be an in vitro test. They are the in vivo test of an antibacterial agent would be to infect a test animal with a bacterial strain, then administer the antibacterial agent to the animal to see whether it will combat the infection. So whenever we are doing a study, this is some kind of cell culture things. This is suppose a cell. We are giving a treatment to the this. This is in vitro. In whenever we are restricting it in the cell culture kind of things, and whenever we are taking it in the animal, then it is getting the in vivo. Okay, so whenever animal is involved or living animal is getting involved, we are calling it as the in vivo. And whenever it is doing in the cell culture or any kind of tissue or cell kind of things, it is related to the in vitro study. Okay, is this clear to everybody? This question, everybody? Okay, let's move to our last question. That which of the following statement is true? Okay, so we have already discussed about the combinatorial study, right? So,
Yes.
okay, so parallel combinatorial synthesis involves the synthesis of a large number of the compounds using the same reaction sequence where there is a mixture of the compounds in each reaction vessel. So is this true or false?
Yes, true.
Okay, so what is a combinatorial study? Let's discuss. A single synthetic route is used with different reagents. A large number of the compounds are produced with mixture of each reaction vessel but a single synthetic route is used with different reagents. In a parallel synthesis, there is only one product per reaction vessel. So here it is using a mixture of the compounds in each reaction vessel. Okay, whereas a mixed synthesis generates several products in each reaction vessel. So as here we are dealing with in parallel combinatorial studies carried out in a specific number of the vessels will produce less compounds than a mixed combinatorial synthesis. So this one is the right option. A single synthetic route is used with different reagents. Okay, the reagents are different and the synthetic route is that one. So we are using a different kind of reagents for that particular synthetic route. A large number of the compounds are produced with mixture in each reaction vessel, but a single synthetic route is used with the different reagents. In a parallel synthesis, there is only one product per reaction vessel, then only one product will be formed per vessel, whereas in mixed synthesis generate several products in each reaction vessel. So as we are dealing here in the case of the parallel combinatorial synthesis, so this is the right option. Okay. Okay, so let's meet you in the next session. Thanks for joining.
Thank you.
Thank
Thank
you,
you.
sir.
Thank you.